The second standard in the nervous system unit covers the way in which neurons communicate with one another, specifically through the use of inorganic salts and concentration gradients. This standard reads, I can explain how nerve cells communicate via action potentials and synaptic transmission, and it covers section 10.6 of your textbook. So this heading and slide is the same. The nervous system is responsible for detecting changes, making decisions based on those changes, stimulating your muscles to act, and maintaining internal homeostasis. In the last video, we discussed the structure of neurons and the ways in which we classify them. We also discussed neuroglia. Your first new heading for today is NERVE.2, the synapse. And I would like you to draw this zoomed in picture that you see down here, and next to it, label the steps of synaptic transmission. So the synapse is the region in between neurons where communication occurs via small chemical messengers called neurotransmitters. You've definitely heard of some of these, like serotonin, dopamine, melatonin. There are a lot of neurotransmitters and drugs that affect neurotransmitters that are talked about every day. So these little blue chemical messengers are capable of moving from one neuron to the next, and they are used to communicate. The presynaptic neuron is the neuron that's sending the impulse, and since this is a message that has gone away, this is the axon of that neuron. The postsynaptic neuron is the one that's receiving the impulse. So it's the one containing all of these receptors waiting to bind to those chemical neurotransmitters. Okay, how do we send this message? So first, there is a message that has already been sent from the soma of a previous neuron and is reaching the end of its axon. Let's put them all. When that happens, there are always vesicles, little bubbles made of the same structure as the cell membrane, that are floating and waiting to release the neurotransmitters they hold. When they receive a message, that's what stimulates them to float to the end of the axon, and as they do, they release their products via exocytosis. So that's a chemical, biological process where vesicles release their contents. So the message forced that bubble to the edge, and the way that that happened was through a helper ion called calcium. So when the impulse reaches the axon's terminal, calcium floods in, and that's what guides those vesicles the final distance to release their contents. So calcium is extremely important in nerve cell communication. After those neurotransmitters have been released into that space, the synapse, they will bind to receptors on the postsynaptic neuron, and that will excite and create an impulse down the dendrite to the soma of the next neuron. So again, we had impulse, then in came our calcium, released the contents via exocytosis, binds to receptor, send new message. All right, so that was in between two neurons, but how do we send a message down a single neuron? Your new heading here is cell membrane potential. So neurons are said to be polarized, and what that means is that the cell membrane of a neuron has an unequal distribution of charges inside the neuron and outside the neuron. So there are these ions that are floating around in unequal quantities. And sometimes a neuron is called a salty banana. And so what do bananas have inside them? Well, they have potassium. And then which positive ion is in salt? Well, salt is sodium chloride, NaCl, but we're not going to worry about the chloride so much. We're going to focus on the salt. So neurons are salty bananas. Sodium on the outside, potassium on the inside. Overall, the imbalance causes the inside of the neuron to be negative and the outside to be positive. So there are some other things going on in here that cause the banana to be negative and the outside to be overwhelmingly positive. The resting membrane potential, when a neuron is not communicating any sort of message, is about a negative 70 millivolts, meaning that the inside is negative 70 when compared to the outside. The last character we need to set the stage with is the sodium-potassium pump. There's a protein on the edge of the neuron membrane that is responsible for putting everything back after we send a message, and so that will reestablish the salty banana. So it'll put sodium outside and potassium inside. But what are the steps? Okay, an action potential is the sequence of events that occurs to send a message down a neuron. Here's our neuron. It's our salty banana. We have sodium on the outside and potassium on the inside. This is just showing that it's on both sides of the neuron. I'm going to put all the steps up so that my pen is easier to access. So first step, depolarization. So there are voltage-gated ion channels, meaning there are little doors right here. And when they're stimulated, because either a neurotransmitter has stimulated them or another neuron has stimulated them, they open up and all the sodium that was outside rushes in. And so now we have depolarized. If we were negative in here before, we just got a whole bunch of positive ions. And so our charge, which was at negative 70, shoots way up to a positive number. In response to that, because the inside's so positive now, repolarization occurs. So that's when different voltage-gated channels open up and they push potassium out in an attempt to reestablish that negative 70 resting potential. They tend to let too much out, and so you have a moment of hyperpolarization where you're beyond negative 70, and then eventually you take time to return to that resting potential, and that's called the refractory period. During this time, that protein that I was telling you about, the sodium-potassium pump, is trying to fix this mess right here. So we don't have a salty banana anymore. We have banana salt. And so the protein's job is to push everything back to its original side. This takes energy, particularly in the form of ATP, and it takes a second. So that's why neurons have a refractory period where they can't send any new messages. Neurons are said to have an all or nothing response, meaning that there's a threshold of negative 55, and if enough sodium ions enter into the cell to reach that threshold, it will fire the same exact strength of an action potential every single time. You'll go through the steps of depolarization, repolarization, and hyperpolarization to the same degree, to about positive 30 millivolts. If you don't reach that threshold, like it says over here, no action potential will occur at all. So that neuron is not stimulated enough to send a message. If all impulses are the same, then why do some feelings like pain, why are they so much stronger? Why is high pressure feel different than low pressure? The answer is the frequency with which we send these messages. So if there is a constant stimulus or a painful stimulus, we'll go through all this and then we'll go through it again really quickly, and then we'll go through it again really quickly, and those should be the same height, but really quickly, versus less stimulating actions will happen slowly.